Hi friends, in this video let us understand where the genes are located and the scientific evolution of definition of gene through seminal research papers from 1953 onwards. Let's begin with where are genes located. Genes are located inside each cell primarily in the nucleus, even though there are some genes in mitochondria or chloroplast in the case of plant cell. As you see, this is the nucleus. Inside the cell nucleus is the site where genetic material is present, and we can see genes in different forms during different stages of cell division. In a resting stage, we see it as a thread-like structure which is called as chromatin reticulum. When at the time of division, this condenses and forms chromosomes that is visible under a microscope. So this is a chromosome. Chromosome is a condensed form of chromatin that is made up of DNA, protein, and some amount of RNA. So when we decondense this chromosome, we'll be getting the double-stranded DNA. And within that DNA, as you see, there are two strands. The backbone is the same, which is made up of sugar and phosphate, and these four nitrogenous bases, A, G, C, D. This changes in bases, and these are the letters of life that codes for specific proteins. It was actually Francis Crick who proposed the central dogma of biology, or flow of biological information. In 19 September 1957, Francis Crick, as part of Society for Experimental Biology Symposium on the biological replication of macromolecules, held at University College London, he presented a paper. For the first time, the idea regarding the flow of biological information. And this was his opening statement. I shall argue that the main function of the genetic material is to control, not necessarily directly, the synthesis of proteins. There is a little direct evidence to support this, but to my mind, the psychological drive behind this hypothesis is at the moment independent of such evidence. At that time, we don't know much about how this gene codes and how it forms a protein, and he was the first one to suggest this, but without an evidence. It's a note made by Francis Crick regarding how the flow of information happens. As you see, he draws arrows from DNA to RNA to protein, or DNA directly to protein, but he was quite sure that the reverse thing never happens. That is, proteins will not form the DNA. Once the amino acid is formed, it is stable and it cannot form or reverse back to form RNA or DNA. And this is the basis of central dogma of biology proposed by Francis Crick. He proposed that DNA can replicate. It's a self-replicating genetic material. Then upon transcription, it forms RNA. And upon translation, the codes on DNA decides the codes on RNA, and that ultimately decides the amino acid sequences within a polypeptide or a protein. So from this, we can define gene as a segment of DNA that codes for a functional protein or a polypeptide. But ribosomal RNA was discovered by Palade in 1954, and he was awarded with the Nobel Prize later. So we know that there is ribosomal RNA. It was called as microsomal structures, but not exactly the function. Function was not known at that time, even though ribosomal RNA is known. Then later, tRNA was discovered by Paul Samnick and his team in 1957 that mediates the translation of information from mRNA. tRNA was the first non-coding RNA to be discovered actually. Rather than mRNA, there are genes that codes for specific RNAs with specific functions like tRNA. With that discovery, the definition of gene has further changed. Gene codes for not only protein, it can directly code for ribosomal RNA or that makes the ribosomes, and also tRNA that is involved in translation. Now the definition has changed. A gene is a segment of DNA that codes for a functional protein or a polypeptide, tRNAs and rRNAs. Then once again, a seminal discovery. Thomas Seck and his team discovered RNA enzymes which are called as ribozymes or RNA with catalytic activity. The term ribozyme was first introduced by Kelly Kruger in a paper published in Cell in 1982. And this discovery further expanded the roles of RNA molecule inside the cell. Rather than just an intermediate between DNA and protein, and there are many RNAs with specific roles, even RNAs with enzymatic activity just like proteins. And this is a paper published in the journal Cell in 1981 regarding ribosome or RNA with catalytic activity. There are many ribosomes discovered like spliceosome in mRNA processing, then peptidyl transferase that is involved in translation, telomerase that is involved in telomere replication, all are actually RNA molecules with catalytic activity. Now we have this definition. What's a gene? A gene is a segment of DNA that codes for a functional protein and different types of RNAs like tRNA, rRNA, ribozymes, snRNAs, etc. Hope you are clear. Take care, stay blessed. Thank you so much for your support. You are with biologyexamsforyou.com.